In this final part, we focused on SBayesRC, a method that I recently developed in our lab to integrate functional genomic annotations into polygenic prediction. Functional genomic annotations provides almost orthogonal information that is potentially useful for polygenic prediction. And the functional genomic annotation can include chromatin states, biological function of the sequence, and molecular QTL information. While LD help us to tag the causal variants, annotations can help us assess whether a SNP is biologically plausible as causal and can potentially inform how should we weight these variants in the prediction equation. Here we show empirical evidence that functional annotations are informative. As shown by our own study and also other studies, the SNPs are enriched in the biologically important annotation for SNP-based heritability. For example, the SNPs in the conserved region or enhancer and promoter regions tend to explain more heritability on average. This motivates using annotations not just for interpretation, but directly in prediction models. The rationale is if an annotation is enriched in genetic signals, it should also help with genetic prediction. Functional annotations in general can inform two aspects of genetic architecture. First, the probability that a variance is causal can be different between annotations. And second, the distribution of the causal effect size can vary across annotations. A good model should capture both dimensions simultaneously. And there is an important challenge when the causal variants are not directly observed. LD allows SNPs to tag causal variants, but the annotation may not transfer or shared between the causal variants and the SNPs in LD. So this mismatch may cause a problem and motivates the modeling of all SNPs jointly in the model and allow the annotation to modulate prior beliefs rather than hard filtering. In the literature, there are several existing methods attempt to use annotations. However, most of these methods only focused on HAMAP3 SNPs and do not use annotation jointly with the GWAS data. So most of the methods, they take stepwise analysis, first estimate the enrichment of heritability in the annotation, and then use as a prior to inform SNP effect estimation. We think there is a benefit to model both sparsity and the effect size heterogeneity in a unified single-step framework. So this leads to two key gaps. First, we need a model that can fit all SNPs simultaneously while incorporating functional annotation data. And second, this model should allow variation across SNPs in both the probability of being causal and the effect size distribution. These gaps motivate the development of SBayesRC. So the core idea of SBayesRC is to extend SBayesR by making the mixture prior SNP specific. Instead of using a single mixture distribution for all SNPs, the mixing probability here would depend on the functional annotation at each SNP. This is achieved using a hierarchical prior where the annotation influences the mixing probability via this generalized linear model. And we estimate all parameters, including the annotation effect and the SNP effect from the data. Essentially, what this model says is the functional annotation will help the model to make the decision which mixing component the SNP effect should go into. If the SNP carries the important annotation to the trait, this annotation will jointly increase the probability of the SNP being causal with a large effect size. And as a result, we can weight each SNP differentially according to their annotation, and therefore improve the prediction accuracy. So there is benefits of using this generalized linear model in SBayesRC. So first, this framework provides can naturally accommodate binary annotation and continuous annotation, as well as the overlap between annotations, as such overlap is quite prevalent in human genome. And second, we estimate this annotation effect jointly in this generalized linear model. So that facilitates the interpretation as each annotation effect is conditional on the presence of all the other annotations. However, this model also present some challenges. As you can see that if we have 5 pi values, then we need to have 5 sets of annotation effects. And these pi values need to sum up to 1. So it's difficult to sample the annotation effect without constraint. Our solution is to reparameterize the annotation effect model to to a sequence of independent binary decisions. Specifically, instead of putting the annotation on the pi values, we construct a set of two-component independent models. For all SNPs, we ask whether the SNP should have a zero effect or non-zero effect. And this P2, this is the probability that the SNP has non-zero effect. And for the SNP with non-zero effect, we further ask whether the SNP should have the small effect size or at least a moderate effect size. And this P3 is a conditional probability given the SNP is non-zero. What's this probability to have a non-small effect size? So similarly, for the SNP that passed the threshold of non-small effect size, we will ask whether the SNP has a moderate effect or large effect. So again, this P4 is a conditional probability given the SNP effect size is non-small. So you can see that we now construct a set of equations and with this P as the conditional probability, and they are independent of each other. Then we are ready to put this annotation effect on the conditional probability P rather than pi. And this is the straightforward to do. Conditional annotation effect, we can compute this P probability. And we can also map the conditional probability back to the joint probability pi using this simple expression. And we assume a normal prior distribution for each annotation effect. So that enabled the Gibbs sampling for all of the parameters. So we can implement the SBayesRC as a full Bayesian method and scalable to the genome-wide analysis. Here I'm using this toy example to illustrate how annotation can translate into the SNP-specific prior mixing probability. So this is our input data for annotations. Rows are the SNPs and column are different annotations. We always include the column of 1 as the intercept in our generalized linear model. And this annotation, we use 1 or 0 to indicate whether the SNP is within the annotation. But you can also use the continuous values for an annotation. So this annotation matrix will be multiplied by the annotation effect matrix, which is estimated from the data, to give the prior conditional probability, P. And this conditional probability will be transferred into the joint probability pi. And as you can see that depending on the annotations, different SNPs would have different mixing probability pi. And some SNP would, a priori, has a higher probability to has the large effect. So this is how the annotation can inform different prior mixing probabilities. And given these different prior probabilities, as an outcome, we will have a different prior distribution for different SNPs. So depending on these pi values, even though each of the components is the same normal distribution, a combination of them would produce the different prior distribution for the SNP effect for different SNPs. And these SNP distribution is annotation dependent. So this is a key distinction between SBayesRC and the other annotation agnostic methods. Another feature about SBayesRC is we developed this low rank modeling. This will allows us to fit many SNPs at the same time. And in our recent study, we fit 13 million SNPs at the same time in the model. So this is how it works. First, we partition the genome into quasi-independent LD blocks. And in each LD block, we decompose the LD correlation matrix into the matrix of eigenvectors, eigenvalues, and the transpose of the eigenvector matrix. Now here's the trick. Instead of using all eigenvalues and eigenvectors, we only use the top ones that explains most of the variation in LD. And if we do that, then this becomes the rectangular matrix. And if we multiply both sides of the equation by the selected eigenvalues and selected eigenvectors, we will end up with this new set of equation. And in this set of equation, the response variable would have less elements than the original GWAS marginal effect. And in fact, these new sort of data points, they are collapsed from the GWAS marginal effect such that they are independent of each other. So the residual are independent. And also this new incidence matrix Q has the rectangle form. We're still estimating all of the joint effect, but using a less data points. So this not only improved the computation efficiency, but also improved the robustness of the methods, because now we are able to sample the residual variance. And you might wonder how much PCs we need to keep. It turns out that we can throw away 80% of the PCs, only use the top 20%, and those are adequate to explain 99.5% of the variance in LD, at least in the European ancestry. So as I mentioned, this low-rank modeling not only improved the computation efficiency, but also improved the robustness of the method. We test that using a smaller LD reference sample size, or in an extreme case, use LD from a different ancestry. As expected, the prediction accuracy reduced. However, we will be able to preserve the higher proportion of the prediction accuracy compared to the other methods. Another common problem in the meta-analysis of real trait is that not all of the SNPs are genotyped in the subcohorts. So as the higher proportion of the SNPs are missing from the subcohorts, we observe the reduction in the prediction accuracy. However, again, we'll be able to preserve the higher proportion of prediction accuracy compared to other methods. And also, we tested the methods using the FinGene GWAS data, where the LD reference is from the UK Biobank. And as the FinGene is the relatively isolated population, there is a LD difference between FinGene datasets and the UK Biobank European ancestry. As a result, the other methods tend to have a decreased prediction accuracy, where we are still able to improve to some extent for different traits. So we think that these all thanks to the low rank modeling, because removing the small eigenvalues make the models more robust to the LD variation between samples. To evaluate the performance of SBayesRC, of course, we did the simulation and lots of real data application. For the interest of the time, here I only demonstrated results for chance ancestry prediction. And here we compare to the method called the PRSCSX. This method doesn't use function annotations. Instead, it combines with another GWAS dataset of the target ancestry. So in this analysis, we are using the UKB European GWAS to predict the UKB East Asian samples, and also incorporate another GWAS dataset from Biobank Japan. And when we do that with PRSCSX, we observed about 17% improvement in prediction accuracy. Now, the question we want to ask is, how important is the function annotation data compared to another GWAS datasets from the target ancestry? So if we only use the European datasets, but combined with but integrated with the function annotations and the high density SNPs, now we observed 16% of the improvement in prediction accuracy. That's already comparable to the results from PRCSX. When we use all available information altogether, that includes the functional notation and also another GWAS dataset from the Biobank Japan, we almost doubled the prediction accuracy improvement compared to PRCSX, reaching to 33% improvement. So this benefit is not specific to East Asian ancestry. We apply the same analysis to the African ancestry and also observed a significant improvement when combining all available source of information. Another interesting finding from our analysis is the significant interaction between SNP density and the annotation information. This graph shows the benefits of adding annotation into prediction at different SNP sets. So the x-axis shows the relative prediction accuracy using annotation compared to not using the annotation at 1 million HAMAT3 SNPs. And the y-axis shows the same thing but at the 7 million imputed SNPs. So if the benefits of using annotation is independent of the SNP set, we should expect to see the results on the diagonal line. But most of the results shows a higher benefits when using 7 million SNPs compared to 1 million SNPs. So this indicates that annotations help more with increased SNP density. And this is not hard to understand. As initially we argue that the SNP markers can tag the causal variants when the causal variants are not observed, but they may not share the same annotation. So that suggests we had better to analyze the SNP panel, including the causal variants, to maximize the benefits of using annotations. And with SBayesRC, we can also evaluate the contribution of annotations to the overall prediction accuracy. And interestingly, we found that the evolutionary constrained regions, although they only take 3% of the genome, they contribute to 40% of the prediction accuracy. And we can also evaluate the per SNP prediction accuracy enrichment. And we found that the non-synonymous variance has the largest enrichment. And this enrichment is aligned with the per SNP heritability enrichment. Beyond prediction accuracy, the SBayesRC also provides insights into the functional genetic architecture for the traits. This is the results across different complex traits. And now we are able to dissect the high heritability enrichment in specific function annotations to the contribution of the different effect size distribution. For example, for the non-synonymous variants, which shows the highest heritability enrichment, we found that most of the enrichment come from the SNPs with large effects, indicating there is large effect and genetic architecture specific to the non-synonymous variants. Last but not least, we show that the SBayesRC can also be used as the genome-wide file mapping tool, and we published this study in the Nature Genetics recently. And basically, we show two things. One is there is a benefit of doing the genome-wide file mapping rather than GWAS loci specific file mapping, because we found that about 30% of the credible sets across different complex traits are located outside of the GWAS loci. And these credible sets can capture, on average, 15% of the heritability captured by the file mapping. So this is not trivial. And also, doing the genome-wide file mapping can be benefited from incorporating functional annotations. So we provide this GCTB software which implement SBayesRC for both prediction and file mapping. In summary, in terms of methodology, we developed methods that incorporates functional genomic annotations to better capture the causal effects and thereby improve the prediction accuracy. We also developed a low-rank algorithm that can fit very high-density SNPs all together in the model, and that improved the computation efficiency and also the method robustness. This SBayesRC methods was developed to use the GWAS summary statistics, but also can apply to individual level data and can be used as a file mapping method. In terms of science, we found that for chance ancestry prediction, using functional annotations provides comparable and additive information to the use of additional GWAS datasets of target ancestry. So it's best to use all available information at the same time. And the functional annotation has been generated, so it's almost like the free resource to use. We also identified a significant interaction between SNP density and the annotation information. This suggests that in the future, maybe using whole genome sequencing variants with annotations can maximize the prediction accuracy. And SBayesRC also provides the natural framework to do the functional partitioning. And our analysis highlights a major contribution of evolutionary constrained regions to prediction accuracy across complex traits. And the highest contribution from the non-synonymous variants for per SNP heritability enrichment, for per SNP prediction accuracy enrichment. These papers describe the SBayesRC methods for prediction, file mapping, and our perspective on the utility of functional annotations for prediction. And in the practical exercise, we will apply SBayesRC to a GWAS dataset step by step. And thanks for watching.